All right, folks. Thank you so much. I'm going to say get in your chairs if you can. We're going to go ahead and get started so we don't stay totally out of time. Okay. So talk louder. You can hear me? I can't hear me. You can hear me. Can you guys hear me in the back seats there? Okay, that's much better. All right. Okay, everybody. So thank you so much for coming back into the room. That's great. As you guys remember, I'm Mindy Buchanan, and today's session runs about 45 minutes. So that will include a question and answer session. Please do save your questions for the end of the session. We'll come around with a microphone. Please do wait for us to get to you with the microphone so that everyone can hear it. Also, this session is being recorded, and the only way your question will be recorded in the recording is if you speak it into the microphone. So make sure that you wait for us to get to you with the microphone, and we'll also help you because it's like a little bit weird. It doesn't like to pick up sound so much, which is weird because that's the microphone's job, right? All right, so I am delighted to introduce you to Dr. Seward Rutkove. I said it right earlier, now I said it wrong. Dr. Rutkove is a professor of neurology at Harvard Medical School and serves as the Nancy Lori Marks Chair of the Department of Neurology at Beth Israel Deaconess Medical Center. Dr. Rutkove's research focuses on application of innovative clinical neurophysiological techniques for assessment of neuromuscular disorders. And he's going to talk to you today about DM research, what you need to know about clinical trials. So welcome, Dr. Rutkove.
Great. Thank you, Ellen. So good morning, everyone. I just want to thank the Myotonic Dystrophy Foundation for asking me to speak today, and it's a pleasure being here and seeing all of you. I would say, in reference to, I forgot his last name, Dr. Robert, the new board member, his comment about having, diseases when you're in medicine. I think I've had ALS at least 3 times in my life now, so I know exactly where he's coming from. Anyway, it's really great to be here, and I'm going to talk about a very general topic today, which is clinical trials. And what I'm talking about isn't really specific to myotonic dystrophy type 1 or type 2, but clinical trials in general. But a lot of the lessons that we have from those, I think, can be very informative to what to understand and expect if and when you participate in a trial of myotonic dystrophy or one of your loved ones or friends do. These are my disclosures. It's not really relevant to anything I'm talking about here today. So the first question is, what do people mean by a clinical trial? And that's not always 100% clear. It could mean simply clinical research. And if you're kind of a little sloppy with your terminology, as a lot of people are, including myself sometimes, it could just mean a trial, a study assessing disease progression, or maybe a new diagnostic test technology, which is something I spend a lot of time working on, or a new quality of life measure, for example, or maybe assessing a new questionnaire. So there are a lot of things that could be considered a clinical trial, but generally those are called clinical research and not the clinical trial per se. It also could mean evaluating whether a new therapy works or not. And that's what would be considered a true clinical trial. Does it work? Does it cause any harm or have any other adverse side effects? And if there's some unexpected effects, maybe good, maybe bad. And so that's typically what we talk about when we're thinking of a clinical trial. And clinical trials in general are divided into 4 phases. So if you heard the term phase in a clinical trial, don't get confused. They're not talking about phases of the moon. It's just basically, and the moon is, of course, very apropos in this coming weekend, or Monday, I should say. But rather sort of different stages or different categories of a part of a clinical trial as we move a potential therapy, or sometimes a diagnostic, more often a therapy, towards approval, FDA approval eventually. So I will work through just briefly the 4 phases so you're familiar with them, and then we're going to get into some more details of what an actual clinical trial looks like. So if you hear about a phase 1 clinical trial, and I know there's at least one company here today that's is looking at working on starting a phase 1 trial in myotonic dystrophy, it's really a question. The idea behind a phase 1 trial theoretically is safety. is the drug safe? So if you hear a phase 1 trial is done, the best outcome they can get is that the drug is safe and doesn't have any serious adverse effects that they were not aware of. Sometimes phase 1 studies will include only patients with a condition, but sometimes they may include actually healthy individuals, just to see how the drug is flowing through the body, if there are any effects, whether it gets into the brain or the muscle, that kind of thing. So phase 1 are just sort of learning about the drug and whether it has the potential for working. But here's the thing about phase 1 studies. The drug company, the researchers, they're actually secretly wanting to see whether the drug's working or not too. So then it's not secret, but they're trying to get some early information to say, well, if it is is there any evidence that it might be having an effect? And if there is, then that will help guide them in how they design their phase 2 study. So phase 1 studies are really trying to do a lot of things at once. Another really important part of phase 1 studies is that they also they may look at dosing a little bit. And they might try 2 or 3 doses to see which is going to be most effective. So the phase 1 studies set the stage, and that's about all they're geared to do. Phase 2 studies are really where you're looking for that first evidence of efficacy. Is the drug really working? And the question is, what does working mean? And I'll come back to that a little later, but is it going to relieve or reverse or stop the progression of the condition? Is it safe? So this is a much larger study. These are typically done in maybe 50 to a couple hundred people, maybe 300 people, in which you're trying to really get a sense as to whether or not the drug is going to potentially work in a larger population. And, and maybe there still will be a dose question there. Maybe they got it down from 4 doses to 2, but they're still going to include 2 doses in this part of the study, in this phase of the study too. So a good phase 2 study, if it concludes well, would be it showed an effect, and it also showed that it was relatively safe with few side effects. And that would be a great outcome from a phase 2 study. I would say also that one does these studies often in a blinded, randomized fashion. I'll come back to talk about what those terms mean in just a couple of minutes. Confirmation is really phase 3. And the phase 3 studies are the critical studies that the FDA looks for to sort of prove that the drug is going to work, it's safe and effective, and actually does what it says it can do. And these are big studies, typically, and several hundred people to several thousand people, depending on the nature of the disease and how common it is. And it will have a very specific predefined outcome measure. I'm not going to get into that too much, but is the outcome measure a questionnaire? Is it proof that muscle strength, for example, is better than people who didn't get the drug? Is it showing that people live longer? That's like, for example, in ALS, where survival with the disease is actually one of the major outcome measures used typically. And then does it interact with other medications as well? So phase 3 studies are sort of the big studies. If you have a positive phase 3 study, the drug works, it met its endpoint, a primary endpoint, as they say, and it also was safe, then you can go to start thinking about going to the FDA and asking for approval or for consideration of approval. I'll come back a little bit about the FDA approval process later. I'll just briefly mention phase 4 studies. They're sort of follow-on studies. Often after a drug is approved, people will continue to identify whether the drug is working and is safe. Sometimes you don't see a significant side effect until thousands and thousands of people have been taking the drug, and you see far out, there's some maybe years out, you start to see that there's actually a side effect that no one had identified before. Or maybe it doesn't work as well in one subpopulation of people that you might not have ever identified earlier. So phase 4 studies don't worry about phase 4 studies. They're out there, but not relevant right now. So I think phase 2 and 3, phase 3 being sort of the one that you really want to win on. Some studies have A's and B's and maybe even C's behind them. You'll see one, phase 2A study. I don't need to get into this too much. Just suffice to say, this is sort of a self-descriptor to state that the study is looking at at some other questions, or it's earlier in phase 2 versus later than phase 2. Like a phase 2A might include a couple of doses. A phase 2B study might then be a continuation with only a single dose in a given disease. So it's kind of how they play around with the letters a little bit, just tries to help define things a little bit. And phase 3 studies, it might, at least as far as I know, usually don't have letters attached to them, but anyone disagree with that? I think I've seen one. Okay. Okay. So now I'll go through some terminology, which you'll hear about, and maybe you know about, maybe you don't. Blinded versus unblinded versus open label, single versus double blinded, randomized and unrandomized, open label again. There's a lot of open labels here. That was obviously a mistake. And lead-in period, and finally dose escalation. I'll talk about all these just kind of briefly to give you a sense of what these things mean. So blinded versus unblinded and single versus double. Sometimes you'll hear these as masked or unmasked. Blinded and masking is all kind of the same thing. And unblinded is also sometimes called open label. So these terms are kind of kicked around a little bit. Some are a little bit more European, those European folks we were hearing about earlier. And in the US, we tend to really say blinded and unblinded. So blinded means you don't know what you're getting. And often the doctor doesn't know what they're getting. So you register for the study, you get enrolled. I'll talk about that in a minute. And then you get handed a medication, and you don't know if it's a medication or it's not a medication. It may be a placebo. And that's a typical blinded study. In an unblinded study, you know you're getting drug or you're not getting drug. And those are typically the terms used for that. Most of them tend to be double blinded. Not only does the patient not know, that's the patient, presumably. But the physician also doesn't know what you're getting. So no one knows what's going on. Somebody does, fortunately, and it would be really bad if no one knew what's going on. I've seen sort of that happen one time, but it involved mice, not humans. So that's okay. The idea here is that no one knows, so that makes everything much more honest and effective. No one's kind of being convinced that they're on the medication because they know they're getting better because they know they're on the medication. Sometimes, I'll come back to this. I don't know if I have it coming up. I think I have it coming up in a minute. And the term randomized and versus non-randomized is also important. Most studies that we talk about are randomized. It's very unusual to hear about a non-randomized study. That means when you walk in, it's a coin toss as to whether or not you're going to get the medication or the placebo. And I'll talk about that in a little bit more detail in a second, but you don't know what you're getting. By the way, in case you're interested, I was, of course, looking for figures for this talk, and I found this recent article on coin tosses, okay, which you think it's a 50% chance you're going to get heads or tails, right? They use it in sports games. Ends up, it's about 51% of the whatever's facing up is a 51% chance that it's going to actually land coming up. So you always go with whatever's sticking up for the coin toss. Just hope that's useful information. Non-randomized are actually, you kind of know what you select certain people for certain medications. And that's not typically, or whether they get a medication or placebo, not typically something you would do as part of a clinical study we'll discuss here. So in general comments on trial design, first studies with a new drug are typically open label. And as you move into phase 2, studies become blinded and randomized, but there are plenty of phase 1 studies that are also blinded as well and randomized. Phase 3 studies are almost always double blinded, randomized, and placebo controlled. There are occasional exceptions where you can't blind the patient to the intervention. So one example is like anything with a surgical intervention, it's really hard to convince the patient they didn't have surgery, right? So, hopefully. So in those, the physician or the researcher doing the study may be blinded. Like the person will come in, they've had surgery, but they're wearing clothes. They can't know that they have a scar on their abdomen or their chest, and the patient doesn't tell them whether or not they received the therapy or not. So the clinician making the assessment, the investigators making the assessments don't know whether or not they were treated. So that's a single blinded study. Those are pretty uncommon too, certainly in the pharma world, the drugs that were probably relevant to DM1, probably not a common situation. One thing that's always worth knowing about is double-blind, placebo-controlled, randomized study wasn't in the Bible. It wasn't in the Constitution. It was actually only developed in the mid-20th century first proposed by Austin Bradford Hill, who was a statistician in England. And this was actually the first double-blinded, placebo-controlled, randomized trial was actually a trial of streptomycin for treatment of TB, published in 1949 in the British Medical Journal. So really, it's not that long that we've been doing placebo-controlled trials for. And this is people who do clinical trials know of Austin Hill, but he's somebody who who people should know more about because he's really changed the way medical science is done for the past 70 years now. Another term you might occasionally hear in certain trials is what's called a lead-in period. So rather than just starting the trial and being assigned to drug or not drug, you actually you actually might get enrolled into the study. And then when you get enrolled, rather you'll do assessments for a period of time prior to actually starting to get medication or placebo, one or the other. And then you're followed over a period of months on both. And this is sometimes you'll hear about lead-in designs. Why would you do a lead-in design? Just to sort of delay things for no good reason, to torture people while they're waiting to get the potentially effective medication. The reason is that sometimes the data is tricky, and getting a single like baseline measurement before when a person comes in may not always reflect the total state of the disease in that person. There's some variability in the measurements. Maybe the patient wasn't in a great mood that day, and so that adds some additional noise to the data. So ideally, if you get a couple of measurements at a couple of different weeks or months apart, you're going to show stability. In certain conditions, I think probably less so in myotonic dystrophy. I don't want to speak too offhand because I don't know what's going on out there in terms of leading designs. But in ALS, for example, which is a much more rapidly progressive disease, I mean, it's a really rapidly progressive disease. If you can track people over a period of months and understand how rapidly or not rapidly they're deteriorating. And then when you start the drug, you can see whether or not that is starting to change their trajectory of disease progression. So lead-ins occasionally occur. It's a good thing to know about. May not be that relevant here. Dose escalation is just another term you may hear. It just means they start at a low dose. They want to make sure it's well tolerated. And then over time, the same people may increase the dose if they're still tolerating it. Or they might be, the first group starts with a low dose. Then a second group comes in and goes on a somewhat higher dose, and the third group to a higher dose. And if they're all tolerating it, all goes well, you can then maybe move into a bigger trial with a single dose going forward. So dose escalation, just another term that you might see. So I will have a sip of water, and I will get into the next part. And my stopwatch stopped. Oh, there it goes. So what to expect in a clinical trial? Well, clinical trials have a lot of parts to them, and a lot of it I'm not even going to talk about, which is like under the hood. Beth is Deaconess. Thanks, Thurman, for the plug. Appreciate it. And my clinic, we're going to be participating in the avidity study, which hopefully will be starting later this year. And so we've been already talking with avidity and the CRO they're working with for the past many months already. So there's a lot that happens under the hood that people don't even know about before any study can get going. And that's just me, not even at the pharma company as they're sort of getting that going. But from your perspective, if you hear about a trial and you say, "Wow, it sounds like I qualify. I don't live that far away from where they're doing it. I'd love to participate." Usually, the first step is obviously reaching out. Somehow you connect up. Maybe that your doctor sends you over there, or maybe you just see about it online, or you hear about it from MDF, as the case may be. You reach out, and you may have a phone. The first thing is usually what they call a pre-screening, and that doesn't even mean you have to come in and talk to anyone. Maybe a phone call, often with a clinical research coordinator or another person at the institution who's doing the study, just to get a sense as to whether you're going to qualify for the study. Things like, oh yeah, do you really have the disease? How do you know you have the disease? Okay, you've had a genetic diagnosis. Okay, that's good. And do you have any major issues going on that might, be a problem? Oh yeah, I've got, advanced kidney disease. That may be an issue. We're not sure you're gonna be able to qualify for it. So we try to get. It's no point in wasting the patient's time and wasting the researcher's time if it's just not going to work, right? So that pre-screening is that first sort of making sure everything looks good, and it sounds like it's going to be promising. Then what typically will happen is we'll invite the patient into the hospital medical center to do that initial evaluation. And this is really what's called enrollment and informed consent. And what's sometimes confusing, what's confused me at least when I first got into this field, that you actually get enrolled in the study before actually being necessary. Well, it's not really, the terms get a little confusing. You sign the informed consent before you actually are necessarily enrolled into the study itself. So you have to go through the informed consent process. And that, again, if you've ever signed an informed consent, informed consents are great. I mean, I have to say, when I first got into the medical field, was first doing clinical research in the 1990s, I would put together a clinical study and I had to submit an IRB application. And the informed consent form was 2 pages. And gradually, over time, just expanded. And when you talk about clinical trials, the informed consents are quite long and quite complicated for a good reason. There's a lot of information. There's safety concerns. There's just a huge amount of challenge that can be associated with understanding them. But informed consents are really important because we want you, the patients and caregivers and families and friends, to feel comfortable with what you're doing and feel free to know that if you do it, it's on your own decision. And most importantly, you can always stop anytime. You can just say, I'm sick of doing this study, I'm leaving. So there's no one's forcing you to do it. But it's all the information is out there. And I can tell you the process of creating an informed consent form is very complicated. So although it may seem like a nuisance, I can tell you a necessity, a huge amount of work goes into these things. So you come in, one of the first things for that enrollment visit, what's really called the screening visit. The other visit was a pre-screening visit. This is the screening visit. At the screening visit, is the first thing you'll be doing is signing the informed consent. And once you've signed the informed consent, it means you're officially now sort of in the study, but not quite, because you have to go through the screening visit. Excuse me. And that means that we've done that pre-screening to sort of make sure there are no major issues, but now we have to go through and make sure you really fit everything carefully. So this is where, as part of screening, we have what's called inclusion and exclusion criteria. And sometimes knowing what's an inclusion criteria versus an exclusion criteria is kind of arbitrary. You could flip them around if you word it differently. But generally, inclusion criteria are what must be true if you're going to participate. So, for example, in a clinical study, you have to be between the ages of 16 and 80. You're 15 and a half, you're excluded. You can't do it. You have a genetically confirmed diagnosis of X, DM1, perhaps in this case. And maybe there's a distance issue that you have to live within 50 miles of the clinical study. I'm just making that up. But because sometimes in some studies, if you need frequent visits and you're getting a very complicated therapy, then you don't want to be living hundreds of miles away or have to fly there to get any evaluation. So those are examples of inclusion criteria. They're not usually a huge number of inclusion criteria. It's a matter of having disease and being the right age. Those are usually the 2 big ones. It's the exclusion criteria that can be quite extensive, and some clinical trials can have dozens of exclusion criteria. And these can be, some really obvious, like you can't be on 2 experimental therapies for the same disease, right? I mean, that would be weird, but I guess possible if they didn't screen for it. Being on dialysis might be one. End-stage kidney disease, liver disease, cancer, smoking too much. I don't know. I'm just listing things, but there can be a lot of different things. Being on birth control if you're of childbearing age, that kind of thing is very, very important. And most exclusion criteria are vetted very, very carefully so that we really get in a clinical trial people who are going to respond and not have unexpected side effects from being involved in the study. So this is really important that we get this right. And so if you pass the screening criteria, it really means you have to be check positive for all the inclusion criteria and check negative for all the exclusion criteria. And if you've achieved that, then you've actually can enter the clinical trial. Now, some of these things are actually not just talking to you and finding out whether you have a history of this or that, or whether you're pregnant, for example, but rather it actually involves getting more data on you before we can actually enroll you. Okay. And I actually probably put this out of place here, but that's okay. So before you can actually be necessarily enrolled, you actually need to get some additional assessments to make sure that you meet all the criteria. Now, in the world of neuromuscular disease and myotonic dystrophy, that may mean some strength testing. Like, if there's a question about does the drug improve strength, for example, you want to make sure the person's a little weak, because if they're not weak, then there's nothing to improve, as one example. Or maybe they're too weak and you're not expecting to have a benefit from therapy. So that could be, for example, they want you in a range of strength. That's just one made up example. Another big one is you usually have a lot of blood taken. That's why I found a picture with lots of tubes. Not enough to not as much as you're donating, but nevertheless a lot of blood, because they want to make sure you have no liver issues, kidney issues, some other things going on. They also want baseline potentially what are called biomarkers or molecules in the blood to make sure that they may be tracking along if you do qualify as well. They'll ask you maybe to fill out a questionnaire. And this questionnaire not is just, I just pulled something off the web, but this is like a standard form maybe to assess what condition you are in with the disease and whether it's going to show changes over time as well. And they may do some additional testing. I showed an EMG test here, which hopefully isn't part of any study, or maybe MRI. So depending on what you're doing, what kind of procedure you're taking part in, there may be a whole bunch of baseline assessments. Some of these baseline assessments are used to track you over time, and we're going to keep coming back to this data as the study goes on while you're on drug or placebo. But others are actually just to see whether you meet the screening criteria or not. So they're kind of doing two things at once. But presumably, if everything looks good, we can go back to this slide. And at this point, you didn't get in, you passed all the inclusion criteria, none of the blood tests came back weird, and you've now been can be officially enrolled into the study. So that's a huge accomplishment at that point. And it means that you can then actually take part in it. So the next point is trial randomization, which is knowing whether you're going to be on drug or placebo. And I always think of Smarties as a great example of a placebo. I personally love Smarties. But people call placebos like sugar pills, but often they're not made of sugar. But anyway, as the case may be, are you going to get a placebo or you can get the real thing? And that's really the question. Now, in the old days, almost all studies were done 1-to-1. It's the coin toss. Well, a coin toss where the coin is flipped around a bit before you flip it, because really 50%, like there's a 50% chance you're going to be on placebo and a 50% chance you're going to be on drug. But in the past couple of decades, people realized that it's much more enticing to actually join a clinical study if you think there's actually more than a 50% chance that you're going to get drug. So many of the newer ones use 2-to-1 or even 3-to-1 drug to placebo ratios. So that's kind of more like that. And so you're more likely to get drug than placebo in most studies, but it just depends on the design. There's an advantage to doing a 1-to-1 ratio, at least statistically. You can need fewer patients to show it works than if you start making the placebo group smaller and smaller and smaller. But the loss in what they call statistical power is not that great. And so it's okay. People are usually willing to buy that if it means patients get more excited about participating, and it's just easier to enroll people and moves everything much faster. So study life, once you're in the study, gets kind of repetitive for the most part. I just tried to make this look as repetitive as possible here, but you're getting something. I put an injection there on the top, and then you just get the same things again and again and again and again for a period of months or many months or a year or something like that. There's usually other things. There's only so much you can stick in a slide, like phone calls to make sure there's no adverse problems going on intermittently. Maybe there'll be other check-in or emails regarding specific questions. Obviously, if anything happens during the study, you would be alerted, like there's a new effect that's been identified, and then they have to reconsent you if there's some issue with the consent form. So all sorts of things happen. It's not just sort of boredom by any means. It's just that there's a standard protocol that's been created, and it's going to be followed over time. And obviously, one of the things is you're on an experimental medication, so you don't know what actually may happen with that. So that means that there may be other side effects that you might be looking out for as well. Let me go just to a little other clinical trials lingo. There's what's called a Data and Safety Monitoring Board. This is a group of doctors and biostatisticians who are actually separate from the study. Their entire role, they're just sitting out there watching the whole thing from afar to make sure nothing bad is going on, that no one's having bad adverse events. At least one biostatistician and some of the committee is unblind. They actually know who's getting drug and who's not getting drug. So somebody, as I said earlier, has to know that. And so that's actually very valuable. And they will be looking for problems that they have to stop the study because of some unexpected side effect. For example, there's something called early termination and futility. Hopefully that never happens, but I'll explain why that might happen in just a second. And sometimes there's an open label extension. I thought I would just show you one other issue, which is called unblinding. This is usually not a huge issue, but if in a blinded study, sometimes it's really not possible to blind people. Like you go, I know I'm getting the drug because every time I get an injection, I get a fever to 104 degrees. You go, okay, probably not. Or look, my urine turns blue. I know I must be getting a drug. So things like this happen, believe me, and people don't even realize it until afterwards. So unblinding is a real issue in a lot of clinical studies, even when they're supposedly blinded. And oftentimes when the study's over, they will ask the patient, did you think you were on drug or placebo, to see whether they were actually unblinded and called it correctly. Futility and early termination is not something you hear about all that much, but it's a very interesting concept, so I couldn't help myself. So if this is your true disease progress over time, it's getting like some issues getting worse and worse, say it's muscle strength. If at some point you did an assessment halfway through the study, so pretend this is 1 year, and at 6 months you do an assessment, and if you're on placebo, this is what should be happening, because you're just getting worse and worse and there's no medication effect, right? If you're on a really great drug, you might see this at 1 year and you go, okay, this is great. The drug seems to be working. No issues there. And because you're assessing, if you go out to a year, it looks very, very promising. So that data safety monitoring board secretly is looking at this too, because they want to make sure things are at least looking okay. So if they saw this at 6 months, they'd be like, okay, potential therapy 1 looks great. Just proceed. No issues. But potential therapy 2, okay, it's still doing much better than placebo. It looks like it stabilizes. It's not making the person better, but it's stable. Great, just let it go. But if you get to potential therapy 3, this is a problem, right? Because it doesn't look like it's doing anything, right? And at that halfway point, they may go, it's not working. None of the signs are showing anything, because this is what's going to end up at the end. So occasionally you hear about studies being terminated early for futility, because it just looks like it has no prospect of working. Why are we spending all this money and time and effort doing this with potential side effects? So hopefully never going to happen in this field, but it's something to know about. Another more common situation is what's called an open label extension, where everyone goes on, you're split initially, but then you go on drug versus placebo. At some point you're on drug versus placebo initially, but then you go on to everyone goes on drug, and you can follow them out. And that's what's called an open label extension. Then many months later, there'll be an announcement of results after the study's over, and the time between when the study's over and it could be really, really long. And eventually you'll hear top line results as to what happened. And usually the company will let you know, and the researchers will let you know as well. So I just quickly here, one question is always, what's the study trying to prove? Is it less fatigue, for example? Is it a symptomatic effect, less fatigue, decreased myotonia, or maybe better sleep? We're more interested in this field, I think especially right now, is drugs that are actually modifying disease. In other words, untreated, this is where things are going to go over time. I love this cartoon, I have to say. I don't know where I found them. Anyway, your muscles are getting better over time, and that's really what you want. Just a couple comments on clinical trials and FDA approval. Does a successful trial equal FDA approval? Sometimes and sometimes not. Strictly speaking, you need 2 positive phase 3 trials to demonstrate drug effect and to presumably show no adverse effects. And one example would be Riluzole back in the 90s, succeeded in that and became an approved drug for ALS. But it is not hard. It's not a hard and fast rule, and especially for harder to treat drugs, sometimes a single phase 3 may do. And then more recently, the FDA got even more looser and goosier about it, and said, well, a good phase 2 study may be enough to get FDA approval. But that may not have been such a good idea. And I think ALS is just it's an important story to know about. And you may know about this story. This is Relyvrio. This was a study in the New England Journal published in 2020 showing a treatment effect in ALS patients of this drug combination of these 2 medications that were used as a single drug. And that led remarkably to FDA approval on a phase 2 study. And then sadly, only this past week, the drug has now been removed from the market because a large phase 3 study was done and showed it had absolutely no effect. So, many thousands of people were taking a medication that just didn't work with side effects and all the rest. So, that's the phase 2 results were real. It was a positive effect in that phase 2 study, but it's called what's a false positive. In other words, it didn't actually work. It seemed to work, but it was just a statistical issue that ended up showing that it did. It's very unfortunate for the ALS community because people were really excited about this. I'm going to just zoom through some of these things in the interest of time. I think clinical trials are very expensive. And I think the bottom line here is really the most important one, that they can cost, a phase 3 study can cost tens of millions, typically does cost tens of millions of dollars. And usually it's through funding from it's the company that's funding it through their own resources and investors, et cetera, et cetera. But sometimes the NIH jumps in and contributes, and philanthropist donors may also contribute in some way as well. Who actually runs the trial? It's actually not often the pharma company. It's often what's called a CRO, or clinical research organization. And I just Googled this morning, and you could see all these clinical CROs just looking for business out there on Google. It's kind of curious. And they do all the details of often finding the sites to do the study, monitoring for side effects, meeting with the investigators on a regular basis, and ensuring that everyone is following the rules. Rules are really important in clinical trials. But I do want to say very clearly, yes, you can trust the results of a clinical trial. As a scientist and researcher and a caregiver and physician, I can tell you that it's really, these things are done really, really well. I mean, these things are policed like crazy, okay? So extremely careful, everything's carefully monitored. The biostatisticians who actually do the analysis are not those ones in the data safety monitoring group. These are people who don't know whether you're on drug or placebo either, and they're just doing the analysis in a completely blinded fashion. So you can trust what's coming out of these things. And also, it's not being directed by the company. It's always being directed by a physician researcher who is not involved with the company. I mean, they may get some consulting fees, but that's about the extent of it. And they take full responsibility for the science that's coming out of it. And again, as I mentioned earlier, sometimes things do, even with an approved FDA medication that does work, there may be other issues that come out as well, including longer-term side effects and such. So just why participate? You want to be one of the first people who are effectively treated, help advance scientific discovery, and for healthy controls, again, mostly in phase 1 studies, maybe perhaps you have a friend or loved one and you feel you can help. And then I think there's also this opportunity to connect and meet with others who are involved, sort of like we're doing here today. I would say it's always good to think before you leap. Many trials are not successful, so participating doesn't mean you're going to be one of the first beneficiaries. You may be one of the first people with an unexpected side effect. So it's always good to remember that. They can be demanding sometimes, a lot of time and travel. So if you're coming into downtown Boston for a study, you got to be prepared for that. There's headaches. We've gotten better at doing more things remotely, but still there's going to be a fair number of visits probably. I mentioned the potential side effects, and I'd say don't sign up for something if you really don't think you can manage it. You don't want to be sort of left hanging. I never know how they get people down off of bungee jumps, but I'm not doing one anyway. One place to find out about ongoing clinical trials is clinicaltrials.gov. If you don't know about it, handy website. There's more in there than you probably want to know. And just final thoughts. I think it's a very exciting time for myotonic dystrophy, obviously. And being part of a clinical trial can mean you're one of the first people to get a new treatment. And it also means you can help advance science, which as a scientist myself, I like, but I think it's another plus. But it does come with some responsibilities and expectations. And just because you want to participate doesn't mean that you'll be able to do to inclusion or exclusion criteria. So always keep that in mind. Just because you're gung-ho about it, there may be some minor thing that ends up pulling you out of the study. And that's just life, unfortunately. And it's always okay not to participate. You have to do what makes sense to you. Don't feel you're obliged to do it just because family or friends are convincing you to do it. You have to really decide for yourself. And my final note is enjoy the eclipse. Yeah. Thank you. Happy to take questions in the short time remaining.
Thank you, Dr. Rutkove. We have a bit of time for maybe like 3 single questions. So if I come over to you and you have 5 questions, you get one of those. I'm going to be clear. And so just raise your hand and one of us will come with a microphone. Oh boy. Okay. Emily, you want to go? Emily's got a microphone as well. So we'll start there. And then, but don't worry, guys, like you will get more time to ask questions because we have what's called Stump the Doctor. So look on your schedule, and that is literally 45 minutes of asking questions. So
just curious, after you mentioned the phase 3 was like the big one, after like a successful phase 3, is there like a typical amount of time it would take for it to become commercially available?
Yeah. So for phase 3 studies, again, as I mentioned, often it depends on what the FDA wants to do. And often they may say, no, the results are great, but not perfect. And you really need to do another phase 3 study. So it could be years from that point. Now, let's say it's a second phase 3 or the FDA goes, no, that's great. We like it. it's usually still going to be a couple of years, months to a couple of years, just because the process, the study's over, the data has to be analyzed, it has to be published in a peer-reviewed journal, and then there's a whole process of going to the FDA and letting the FDA think. There's an advisory council. It's a long process at the FDA itself. So, I don't think there's one answer to that. I would say probably a minimum of 18 months. Anyone on the medical side want to disagree with that? I don't know. Something along those lines. So, sure.
I think I heard in one of your slides there was a picture of an EMG test being done, and you said hopefully that's not being used in a research project. Why is that?
Oh, I'm sorry. Well, I didn't want to. Sorry. EMG is a great diagnostic tool to make a diagnosis, but it's not typically used in a clinical trial because you don't monitor EMG changes over time. Typically, that's the only reason. Plus, having repeated needles in you is not necessarily the most enjoyable. But it is actually there may be approaches to doing EMG as well and getting useful data out of it. That's all.
I just have a question. How many drugs are in clinical trials, and what phase are they in?
Oh, now that I hesitate to answer that question. Does Thurman, do you know the answer to that offhand in my time? So 3 to 4 in what I mean, Avidity is a phase 3. The others are phase 1, or is Dyne is. Oh, there's a list.
Oh, yeah. Also at the advocacy table, MDF has a drug development pipeline document that has a little arrow that shows where folks kind of are. It's also on our website. Okay. One more question. Emily's coming to the last person. I promise. Stump the doc.
And he just stumped the doc, by the way, just to be clear.
I don't think I need.
Well done.
I'm just wondering how they tell if the medication is keeping a patient stable or whether the patient's just stable by themselves.
But that's a tough question. And that's why they do studies with many people in them, right? Because on average, they sort of know what should happen. So if one individual person isn't really changing over time, that's sort of comes out in the wash a little bit, but it's just a series of doing enough people, in general, slowly somebody maybe is getting a little worse every year. Overall, they'll pick it up much more easily than if it's just in one person. Great. Well, thank you all very much. Have a great rest of your day.
Thank you so much, Dr. Rutkove. Thank you.